So hello everybody, my name is Wolfgang Hankeln and I'm sitting here with Natalie Simon from Roscoff, France. And today we are going to talk about the GSC14. Could you please introduce yourself and talk a little bit about the fields of research that you work on?
Yes, so I'm working in, I'm both doing some research and some teaching in the field of marine ecology and marine biodiversity. So I'm mostly, my research focuses on photosynthetic organisms, marine photosynthetic organisms, and especially phytoplankton. But I also teach all kinds of, I teach about all photosynthetic organisms, including macroalgae. And in terms of research, my research focuses on the ecology and systematics and taxonomy of phytoplankton.
So this is now the 14th GSC meeting. Which were the topics that were particularly interesting? So, and why did you attend this meeting?
Well, I did attend this meeting because for my research and also for the research we are doing on our observatory site. We are using mostly, classical techniques to study the diversity of phytoplankton and understand the ecology of the main species. But we're also using some techniques using molecular biology, and this for more than 20 years probably in the lab. But we would like, there are some questions to which we cannot answer using our classical techniques, right? For example, when the species we're focused on are morphologically really identical, so then we need other techniques to decipher the ecology of each of the genetic types. And in that respect, we started on our observatory site to sample for, in order to get, to be able to use molecular techniques on these samples. But we did not really implement a strategy to use this sample on a long-term way, right? We just targeted a few questions using a few of these samples. So I did attend this meeting in order to see what was the level of in the international community and to take ideas of how we could use these samples to focus on more general questions on our observatory site.
And what are those more general questions?
Well, about, for example, questions about what triggers the blooms of the phytoplankton species, what is the role of the infraspecific genetic diversity on the dynamics of phytoplankton. That kind of questions are, and also we are in our group on phytoplankton. A lot of us do focus our research on the tiniest phytoplankton species, which have basically the same size as a bacterium, like they are 1 micrometer. And for those ones, for these ones, even in our coastal waters, in the English Channel, probably there are many species that we still do not know, and maybe some key species that we do not know. So, and also, other questions we are targeting are the interactions between all these species, which is difficult to approach using classical techniques.
So this is already the 3rd day of the meeting, it's almost over. What did you enjoy most? What were the most interesting topics?
Well, I did enjoy a lot the discussions that there were, the discussions within the working group on ecology and theoretical modeling. Although I'm not at all into modeling, I really was interested in the way these people, the modelers and the people who are really into this topic could, why they could be interested in such molecular data, and to have a feeling about what could be retrieved from the use of these molecular data using theoretical modeling and theoretical ecology.
And what are the benefits from your point of view taking part in this GOES initiative?
Well, as I told you before, we are already sampling. In our observatory site, we have been sampling for more than 10 years, and we did archive DNA samples for more than 10 years. And I had the feeling that these samples can help us answer some specific questions for our research interest. But I also have the feeling that if we group these samples with samples from a larger geographic coverage, we can answer maybe questions which are more general about microbial ecology and microbial both eukaryotic and prokaryotic biodiversity. So,
And in terms of integration, you already mentioned that there are already existing methods at your research site. Do you think this initiative now, after the meeting is almost over, succeeds in integrating into already existing infrastructures? And are you satisfied with the plans so far?
Well, I think there's a lot to be constructed still, and probably the first step is to bring the people together, and I think when I go back home, I can maybe try to bring back, to bring back the questions that this group have, and to the oceanographers or to the ecologists, microbial ecologists in my lab, and try to have them understand that it would be nice to join this group to maybe improve our protocols, our standards, basically both to answer our questions and to answer more general questions.
Do you think that the OSD is a good way to start this network, or is a good start off for an initiative like the GOES network?
Well, to answer scientific questions, I have no idea. I just wonder if sampling for one day is useful to answer any kind of scientific question. But in terms maybe of bringing the community together and of thinking about protocols, it's really a first step.
And this GOES initiative, do you think, or would you agree that it's also a good way to monitor maybe human impact on the ecosystem?
I think it's a good initiative but I think it's also another way of, another technique to see what's going on in the ocean, and it's got to be added and well integrated with the other techniques which are the classical techniques or the most advanced techniques in other set up by oceanographers to monitor the environment. It's just one more technique which is probably extremely powerful and will be extremely powerful in the next few years. But it's got to be integrated with the others. It's not like it's going to, the rest is going to disappear.
Okay. So thanks a lot for your time.
Thank you.
And enjoy the lunch now.
Thanks.